last Tuesday, I even forgot to bring it in here, and then there was one day in class, I forgot to turn it on, I left it standing over there, so. If I can just get through about 4 or 5 more of these lectures, I'll be done, because I don't have that many left. Anyway, these different types of solutions where we ended up, and those of you that were able to come to class, hopefully you grasp a grip of that, especially when we applied it to real life, when we was out there in the fresh water and the ocean water, and now it makes sense, right? All right, so the rest of this little section is talking about endocytosis and exocytosis, which is things happening inside and outside the cell. Endocytosis, this is where the cell gathers up trash, basically cleans house. And if it's large particles, then it employs phagocytosis, which literally means cell eating in Latin terminology. So it engulfs large particles, puts them if we're talking about animals, which in this class we are, it's a vesicle, would be your trash bag or your trash can. So it dumps these parts in this vesicle. It may be a worn out organelle that no longer works. It could be stuff that shouldn't be in there in the first place, excess something, whatever it may be. If it's liquids and very small particles, then cell drinking takes care of it, or pinocytosis, and again dumps it in the trash can. Moves it out to the cell membrane, where it then kicks it out of the cell, or exocytosis, removes it out of the cell. So it's no more, it's really nothing less than mom gathering the trash in the house and dad setting it on the curb. So this is just how the cell keeps a clean house. If all this trash is rolling around in there and keeps cluttering up the internal part of the cell, then that's going to cause problems. So we keep the cell clean through this process right here. And I usually use literal meanings on these on the test. So cell drinking and cell eating, you'll have it. Any questions there? All right. This brings us over to mitosis, and I do meiosis here as well. It's not, both are not in your book in the same place. Meiosis shows up, I think, in chapter 5 with genetics. But if I wait till then I have to go back and review mitosis again. So I'll do them both together and we get them out of the way. And we can see the comparison a lot better if we do them side by side. So first definition up there, zygote, result of union of egg and sperm. We'll come back and talk about that as we go through. If we do a comparison of mitosis and meiosis, this is just things to remember. Mitosis always deals with normal body cells, everyday normal body cells. When they replicate, they're making more exactly the same. They're cloning themselves. If it's a lining of your stomach, it replaces itself every 2 to 3 days. Your skin replaces itself every 2 to 3 weeks. You're constantly replacing cells all the time. So in mitosis, normal body cells, you have a diploid number of chromosomes. Ours is 46. So represented by diploid, or the 2N representation, in our case 2N equals 46 in humans. Meiosis, always sex cells, always, and we'll do a lot of comparison. In fact, I believe part of the beginning of chapter 5 we talk about the advantages of sexual reproduction over asexual reproduction, because when we get into the single-celled animals such as protozoans, we'll talk about, these different animals use different forms of asexual reproduction. Bottom line, binary fission. So we'll learn in general, when in doubt, just put binary fission and we'll get it covered. But we also know that these animals have the capability of reproducing sexually only in times that they feel the species is threatened. So there are advantages of sexual reproduction. Although we don't produce as many offspring, what we produce, it's better. Okay, so there's kind of a bottom line there. Haploid, meaning half. So what we ultimately have is a 2N cell, a 46 chromosome cell, going through meiosis and producing offspring with half the number of chromosomes. Why is that important? Well, it's as simple as this. If mom has 46 chromosomes and dad has 46 chromosomes, we use this first definition here. Union of egg and sperm, 46 and 46 is 92. Now, how long do you think the human species can continue with each generation doubling the number of chromosomes? So my children would have 92 chromosomes. Their children have 184 chromosomes. I don't even know what that is. I do know what this is. And according to one of our first lectures, this cannot happen. Remember our first lecture that said poodles make more poodles and chickens make more chickens? That is a goldfish. Humans do not make more goldfish. So my point is, without meiosis, we don't sexually reproduce. We have to divide that in half, divide that in half. Look at there, 46. Humans make more humans. Does that make sense? Let me give you a little definition of meiosis here, and then when we look at it here in a minute, we'll actually see this definition applied. Here's your definition. It is a reduction, division, reaction. Meiosis is a reduction, division, reaction. And all that is saying is, you reduce the chromosomes in half, and you double the number of cells. Reduce chromosomes in half, and double the number of cells. See, when we get finished with mitosis, looking at mitosis in a minute, we'll have 2 daughter cells that are identical to the mother cell. When we get finished with meiosis, we'll have 4 daughter cells that have half the number of chromosomes as mom. So reduce the number of chromosomes, double the number of cells. All right, let's take this into the cell cycle. Now I want you to remember this cell cycle, especially interphase, because in chapter 5, when we do replication, transcription, and translation, talk about DNA and RNA, all that's happening in interphase, the cell cycle. So hopefully we can get a visual of the sequential events that's occurring here in the cell. So if I ask you on the test, and I will, what are the phases of the cell cycle? It starts with interphase, prophase, metaphase, anaphase, telophase. In short, IP-MAT. Now that's the cell cycle. Now this is one of those things that there was a day, and it doesn't seem like it was that long ago, that we could not, probably limited by technology at the time, we could not see anything happening in interphase. So the scientists just labeled interphase as a resting stage. Has anybody ever heard that? And that's good to see, because there were several years there that high school science books still had it as resting phase. So it's good to see the books have moved on. And I'll tell you about when this happened. It's somewhere around between 86 and 89. 1986 and 1989 is when they made this discovery. Because I was in college at the time. That was just a few years ago, Katie. Seemed like yesterday, but I don't know what happened in the year between. So once technology came about, and they really got to looking inside this cell, what they saw was all the genetic material was being replicated. We were doubling the DNA. We were making more RNA. We was making proteins. And what interphase was doing is preparing to divide this cell equally and make 2 new cells just like it. We can't just take a cell sitting here and chop it in the middle and say it's identical on both sides, because it's not identical to the parents. Now we have 2 halves of the parent. When we go through mitosis, we have 2 full identical clones of the parent, which means full complement of everything. So the cell cycle, interphase, G1, and every book's a little different here. In fact, if you have your book, turn, oh gosh, it's page 74 in the physiology book, but it's not going to be there. It's going to be back here. It's chapter 3. Right there. What page is that? 53. Look on page 53. Okay, I'll look into that for you. And you'll see a little pie chart there. And the pie chart, all the light blue is interphase. And you see a little purple section there, a little piece, slither of the pie, is mitosis, and the yellow equivalent slither of the pie, which if that was my pie, that's all the piece I would get. It's yellow, and that's cytokinesis, and it happens simultaneous to the mitosis aspect of it, because that's division of the cytoplasm, and we divide it equally as well. So, and what does it say in G1 around that pie circle? What does it say? RNA and functional protein synthesis. Well, this is right off of the physiology book. It says centrioles replicate. I have no doubt that centrioles replicate. And I have no doubt that we have functional RNA and protein synthesis occurring. Do we know exactly at what point and where in this? Probably not. And that's why you'll find discrepancies in these books. I do believe in interphase is where all of this happens. And now what does S say? DNA replication. I think all the books say that. They know that S is DNA replication. S stands for synthesis, for making of. What does it say on G2 on your pie chart? Structural protein synthesis. Physiology book says final growth, and I truly believe that final growth does happen here. I'm not doubting that that doesn't happen as well. We just don't know exactly in perfection where and when it happens, but we do know it happens in interphase. We have to do all this before we start dividing the cell. And so, if I ask you on the test, what phase or what part of interphase is DNA replicated, that's probably the only one we really know, and that's the S phase, right? So, what are the phases of the cell cycle? Start with interphase. Prophase, metaphase, anaphase, telophase. What are the phases of just mitosis? Starts with prophase. Okay, there's your kicker. And for years, interphase was part of mitosis because they thought it was resting. But now we've got a complete cell cycle going, so they've kind of separated things out a little bit. So let's look into mitosis. We looked at these pictures in lab last week, right? The mitosis pictures. All right. So prophase, we have centrioles move to opposite poles, nuclear membrane disappears, chromosomes appear. Metaphase, the chromosomes line up on the equatorial plane. As we saw, it was really organized. So what we're doing in prophase is we're organizing the genetic material. In metaphase, we see it organized. In anaphase, we start organizing the equal separation. Chromatids separate and move to opposite poles, and for telophase, this is where the cell finally divides. But telophase, and this is how the test question is worded on the first test. What phase begins when the chromosomes reach the poles? Telophase. And we can see telophase because when you're looking for telophase, it's easy to see it because telophase is always identified with this furrowing effect. It's starting to pinch apart here. You'll see the furrow. That's an easy one to identify, but at what point does the furrow start? When the chromosomes reach the poles. So that's how the test questions work, right off that definition. Now let's back up to anaphase just a bit, do a little bit of terminology explanation here. Notice it says chromatids separate, move to opposite poles. This is the first place in any of these definitions we've even mentioned chromatids. What is a chromatid? We've heard of chromosomes, right? Okay. A chromatid is a half of a chromosome. If that's a chromosome, each one of those halves are chromatids. Okay. Chromatids are made of chromatin. More confusion. So it's important that we grasp these. We have chromatin, chromatids, and chromosomes. Chromatin is made of DNA and proteins. Chromatids is made of chromatin, but now it's visible. That's all it is. So ultimately, chromatids are made of DNA and proteins, aren't they? Two chromatids become visible, join, makes a chromosome. So what's a chromosome actually made of? DNA and proteins. It's just condensed, cool, organized, and visible. So each stage becomes more visible for us. So those terms you will run across even again in chapter 5, and they just throw them out there without really leading you into what they're talking about. Now, when it says the chromatids separate and move to opposite poles, you should be thinking, well, if a half a chromosome is going to opposite poles, how in the world can that be an equal cell if they just have half chromosome? Well, in metaphase, I think we remember seeing in metaphase that those chromosomes were lined up. We know it's organized. And when they moved to opposite poles in anaphase, we have chromatids moved to opposite poles. These are what's called sister chromatids, also known as homologous chromatids. Homologous means the same. Sister means out of the same gene pool, basically. How many of you have sisters? I know Katie has sisters. Would you consider your sisters to be identical to you? Some of y'all are not even close in being. Some of y'all probably wonder if you have the same father. I mean, that's how different sisters can literally be, and you know you came out of the same gene pool. What ultimately this means is that this chromatid contains all the genetic information that a chromosome has, because this sister and this sister, it may carry blue eyes here and brown eyes here, but she's going to have eyes. That's a positive thing. We may have red hair here and brown head here. Brown head, that's red head, brown head. I don't know. But at least we're going to have a head, right? But the point is, all the genetic material's there. So when the chromatids reach the poles, they're considered chromosomes again. Okay? Now, we're going to apply this even deeper in meiosis in just a minute. So there's a plan here. It's all there. Nobody gets cheated out of anything. We all get our eyes and ears and nose, and if we don't have all the major components, you just probably rejected a long time ago, and you never know it. So, all right. Cytokinesis, division of cytoplasm, happens simultaneous with telophase, and is constantly being divided equally throughout this process. Now this says refer to page, I think it's 99, and I say 99, I don't know in this book. Every time they switch books on me, they change a page. It's still in there.
Well, 99 is just references.
Just references? Okay. Well, 71 is actually the right page in the physiology book. But let me just tell you about P53 and P21. And it's probably in this chapter that we're in right here, if not an earlier chapter. It may actually be in chapter 1. But P53 and P21 are two genes they've identified that actually produces a substance called cyclins. And cyclins, we know, triggers mitosis. So these two genes controls cyclins. And in laboratory animals, what they did is went in and bombarded the P53 gene with X-rays. Now how they were able to just kill that one gene, I don't know, but they did. And when they killed that one gene, cyclins were not controlled. It just constantly produced cyclins. And the more cyclins that were produced, mitosis just kept rolling. Now, we just said a while ago that lining of your stomach replaces itself every 2 to 3 days. Your skin replaces itself every 2 to 3 weeks. And so, with that said, would your skin go through mitosis a lot slower than mitosis of the cells in your stomach? Theoretically, they would. So what we had in all laboratory animals that the P53 gene was destroyed in was cancer. Cancer is mitosis out of control. That's what it is. You get cells in one part of the body that rapidly reproduce themselves to the point they have no place to go, so they slough off in the bloodstream and take off somewhere. They latch somewhere else and start growing there. Is it supposed to be there? Probably not. It's supposed to be over here. So they did the same thing with P21. They killed it. We got cancer. So if either one or both of those genes were destroyed, we did not have any control over cyclins and we got cancer. In every laboratory animal. Now it's nice knowledge to know, we just don't know how to fix it. Now if that was the only cause of cancer out there, we might be able to do something. Well, I mean, we've learned to do some gene replacements, some gene splicing. Who knows, it might be fixable, but that's not the only cause of cancer out there. We bombard our genes, kill our genes by other methods. It's not just radiation. I mean, we all know carcinogens is basically everything you eat, you drink, you're in and around, you breathe. What's not a carcinogen? I mean, if you think about it. And then we take known carcinogens, forms of tobacco and cigarette smoke and chemicals, and stick our hands in it and puff it and everything else and wonder how did we get so sick? And then who knows what else we're exposed to, you know. All right, let's look at these phases of mitosis again. I've got another little here, overhead here. There's an animation plus an actual picture. We have interphase at the top, and if you see that, and your book actually really has good pictures of mitosis, and not bad pictures of meiosis. In interphase, we can see a nuclear membrane, we see darkness and some dark stuff in there, but there's no organization. We can't tell. What's happening is we're replicating everything. The dark stuff is DNA and proteins. We're just not organized, okay? Then we drop off to prophase, and this is an early prophase as we looked at in the lab. Some of the nuclear membranes disappearing, maybe most of it. You see a little more condensedness in the darkness. Well, the darkness are the chromosomes. It's the DNA and proteins condensing, coiling, twisting, becoming more visible. And we move to the next one and we see even more condensed, more organized. Each stage is one step closer to the final product, more organization. We follow that stage down here and now we are organized. There's your metaphase, we're lined up in the center. You can see your centrioles and opposite poles. We come down here in early anaphase, you can barely see light there where we're starting to separate those chromatids, barely. We come over here and we're way separated. We're almost to the opposite poles. What's it called when the chromosomes reach the poles? Telophase. And you can certainly see the furrowing effect up the top of this. We're almost completely finished this one. There's only one stream of cytoplasm left right there. And that's telophase, producing two new daughter cells that are identical to the mother cell. Goes right back up in the interphase. Now do we immediately go into the process again? Depends on what cell we're talking about. Depends on what part of the body we're talking about. Depends on how fast we need to. Depends on P53 and P21. If they're working, doing their job. So this is simple mitosis in the process. Now, let's apply this to the meiosis we were talking about. Use this animation here. All across the top is prophase 1 of meiosis. Now, for us to double the number of cells and reduce the number of chromosomes, we must go through basically the mitotic process twice. Okay? So we have meiosis 1 and meiosis 2. Okay? So at the top, how many chromosomes you see in the first animation? 2 red ones, 2 blue ones? I'm with you. They kept it simple for us. We're just going to play with 4. How many you see in the second one? There's not a trick there. There's still 4. So I'm not sure I can count that high. Still 4, but does the second one look more organized? Third one look even more organized? It does. Each stage we're getting more organized, but there's something happens in meiosis here that's only unique to meiosis, which is only unique to sexual reproduction. The first thing is synapsis. So here's your definition. Synapsis is joining of like chromosomes. Synapsis is joining of like chromosomes. Joining of like chromosomes. So, when you say like chromosomes, you could say sister chromosomes, because it's the same thing. You may not be identical to your sister, but there's likeness. Some there's more and some there's not much, but there's likeness. Now, we join the like chromosomes, but notice the next picture, the blue ones and the red ones start exchanging genetic information. Well, this is called crossing over. So, here's your definition of crossing over. Exchange of genetic information. Exchange of genetic information. You know what's cool about that is we can add diversity to the offspring. You know, where we did have a combination of green eyes and red hair, if we exchange genetic information here, we may have green eyes and blonde hair. We may have green eyes and brown hair. These chromosomes exchange equivalent parts, an eye for an eye, a tooth for a tooth, if you will. That's what they're doing. So when you get here, what do we see? Parts of the red on the blue and parts of the blue on the red, but they still look like full-fledged chromosomes. The color is just indicating we've exchanged those parts. So we make it to metaphase 1. Notice that we don't have single chromosomes lined up, but we have double chromosomes lined up. We have homologous chromosomes lined up, which means we have 4 sister chromatids. We have a big family. We have 4 sisters. Dad probably left out when the 4th girl was born. Y'all don't have that fun. Never mind. This time we actually do move chromosomes to opposite poles. And when we're finished in telophase, we have 2 new cells with half the number of chromosomes. Are we finished? According to definition, we have to double the number of cells, so we've got to go again. So we bring those 2 cells down here to meiosis 2. Not greatly organized, but notice this time we have single chromosomes lined up. This time we're moving chromatids to opposite poles, because here, when we was moving these chromosomes, these chromatids are all sisters. You could mix, match, mingle, however you want to, they're all sisters. So when we move the chromatids to opposite poles here, when they reach the poles, what phase begins? Telophase. When they reach the poles, they're called chromosomes. They're full-fledged chromosomes at that point, because they're all sisters. They'll use different terms for that. They'll call them homologous chromosomes, they'll call them tetrads, they'll call them bivalents, and there's one other term that slips my mind right now, but meaning they're all sisters. Okay? So we look at the last stage here, we're in telophase now, and we end up with 4 new cells. How many chromosomes in each? 2. We have completed our job. We've doubled the number of cells, we've half the number of chromosomes. That's meiosis. Now they say that if an egg is not fertilized at metaphase 2, it doesn't finish. It doesn't need to finish. If it's not going to be fertilized by then, it's not going to be fertilized, it's gone. Are we made to produce 4 equivalent eggs at the same time? You want to have 4 kids at a time? How many of y'all have kids? Would you like to have 4 of the same at the same time? That'd be suicide mom, wouldn't it? And when somebody does, and we hear of it in the news every now and then, somebody's had 5 or 6 or 8, it's like, man, what kind of drugs was she on? Because it took a drug, didn't it? That's not natural, it's not normal. Twins, yeah, we have twins from time to time, but 4 equivalent eggs at the same time is not normal. Now we can promote that. We have drugs that promote that, but it's not normal. So in egg production, when we get finished with meiosis 1 at the top, what we have at the end of meiosis one is we'll have one big egg and one little egg. The little egg is non-functional. It has genetic material in it, just like this one does, but this one stole all the cytoplasm. So both of those two little egg, the little egg and the big egg comes here, so here's a little one, that's a big one, goes through the whole process, and when we're finished, this big egg produces another little egg, and this little egg produces another little egg, and we have 3 little eggs and 1 big egg. That's almost turned into a little joke here. So the 3 little eggs are non-functional. They have very little cytoplasm in them. They have genetics. So what happens when they get fertilized? Absolutely nothing. They die. They probably won't get fertilized. But why do we have one big mother egg here with all the cytoplasm? Because we have to. Because the cytoplasm has the mitochondria in it to produce the energy. So after fertilization, we maintain mitotic divisions from 1 cell to 2 cells to 4 cells to 8 cells to 16 to 32 to 64. If we were equivalent in size, none of them would have enough mitochondria to sustain life after fertilization. So we're made to have one kid at a time. And it's up to that egg to have the energy after fertilization to create life. Never thought about that, have we? Didn't know you was throwing away pieces of eggs every 28 days, did you? Now it's gonna bother you, isn't it? Gosh, dang. I feel like there's 3 little polar bodies just went away right there. That's what's creating the mood swings. You're losing little polar bodies all the time. Ooh, there went a polar body. Not true. Now, what about twins? Anybody have twin sisters, twin brothers, twin brother, twin sister?
I'm a triplet.
And you're a triplet.
Yeah.
Where's the other 2? Did you kill them off somewhere?
No, one goes to school here and one goes back in Arkansas. And then 19 months later, Mom had another girl.
Mom was a slow learner, wasn't she?
Yeah.
She's one tough mother, is what I can say. Willing to try another one. Well, shooting for a boy, maybe?
Yeah.
Okay, so you're having lecture and then you're going straight into lab?
I don't have lab today. I have lab Thursday.
You have lab on Thursday?
Yeah, Thursday we will do lecture at 11 and shoot to lab Thursday.
Okay, the reason why I'm asking is because there's a young lady who needs to ask you a question and I didn't know what time you were going to be out of here.
Shortly after 2.
A little bit after 2?
Yeah.
Okay, because she's supposed to come back at 2. So
Class is supposed to end at 2:15.
Okay,
We usually
2:15.
get out at 2:10 or so.
Okay, all right. I'll tell her hang around.
Okay.
Thank you.
All right, so in twin production, we have identical twins, triplets identical. Man, your mother produced one egg that was seriously getting after some splitting here. Because in identical situations, after fertilization, it splits. There's your identical twins. In her case, it split again.
It was supposed to be 4, she miscarried one.
Holy smokes.
That's not
Well, I've known of twins, be pretty far along and miscarry one, the other one goes on and is fine. Separate placentas, do we technical enough to know if y'all's all in there together? Well, you had to be separate. Because if you lost a sister, then if she was in the same sack with y'all, y'all have been coming out with her. So y'all had to be separate placentas. Well, did you weigh close to the same? Yep, you're all separate. If they was in there together, working in the same placenta, they'd have been fighting worse than they ever did growing up. And survival of the fittest, there'd have been one in there, or maybe 2 in there, might have even starved another one out, unintentionally, but it's survival of the fittest. And they say that at one time, I actually saw a stat. Is she fighting her sister out? I even saw a stat at one time that said 60% of women that do get pregnant start pregnant with twins and never know they lose the second. Now, I don't know where they come up with that information because I'm not sure the human race is fertile enough to do twins 60% of the time. But we obviously don't end up with that many twins. So in an identical situation, egg splitting. After fertilization, the egg splits. Actually, it split more than normal to get 4. And fraternal twins, you have a sister, identical. Then we had 2 different eggs. It could have been a brother. We had 2 different eggs, 2 different sperm. Could have been a brother and a sister being twins. But we had 2 eggs and 6 polar bodies in that situation. So we ovulated a double. And that's not that uncommon. And of course, you'll pass that on to your children. It usually jumps a generation to where your children, your daughters will have a chance. Now, can your sons carry the twin gene? Yes. Are they going to be the cause of twins? No. Their daughters could be carrying the gene for twins. The man has no decision whatsoever in that you're going to have twins or not. Now, he can determine sex, not like it's really his choice, any, many, money, mo. But, you know, you're throwing an egg in and he's throwing 300 million sperm in there. And so, you know, determine the sex, yeah, there's X and Y's in there fighting tooth and toenail to get to the egg. But determining how many offspring, that depends on how many eggs is there. Because if there's 2 or 3 eggs out of 300 million sperm, there'll be enough get there to get it done. So they're fighting tooth and toenail. Now, 50 million sperm is considered clinically infertile. But 50 million, that's plenty. No, not really. So in chapter 7, which is on our final, we'll talk more about that. But that's kind of how this comes about. Now, if this is sperm production, these are 4 equal sperm because we're producing sperm by the millions. Every 60 days, there's a whole new group of sperm produced. I mean, they turn over every 60 days, constantly being produced by the millions. Egg production, normal, 1 every 28 days, 13 a year. Occasionally, more than 1 egg. We might get a happy egg that splits. Any other twins in here?
My grandma had 2 sets of twins.
See, my wife had twin aunts, and she was like, oh, I hope it's not in the genetics, you know. And it wasn't, thank goodness.
Yeah. My grandma had 10 kids, and 4 of them were 2 different sets of twins.
Yeah. Yeah, and I know a set of twins that a few years under me kind of grew up with them, twin girls. And typically it would skip their generation and not them. One of the twin girls has chose not to have children. The other twin girl had her first set of twins. And she said, that's enough. Don't want any more. She's done. Okay, say that till comes along. Yeah.
She kind of tubed after that one.
Yeah, I mean, she is putting corks in it and everything else. Cut it out. Get rid of it. Stop the chances. Any questions over meiosis? And we'll tie into this a little more as we get into genetics as well. Somebody have a question?
That woman who had twins, but one was white and one was black, how did that happen?
She
Well,
cheated on him.
No, well, I thought they
I'm not,
proved it.
I don't guess I'm familiar with the situation, but to explain that, would have to be multiple ovulations. Obviously, they're not identical. And we have to look at the obvious first. And multiple ovulations, and some ovulations can be slightly delayed. There's a short time frame in which conception can occur in a 28-day cycle, and it's about 5 days. 3 days prior to ovulation and 1 day post. So if ovulation of one egg is on day 14 of a 28-day cycle, the other egg could be slightly delayed or slightly ahead of it. So let's say intercourse happened 2 days prior to this egg being ovulated, then intercourse the next day, well, this egg already got fertilized, but the sperm only lives 72 hours. Maybe the sperm from the first intercourse has already died or they're non-existent. And then the next egg is slightly delayed and intercourse puts new sperm in there and fertilizes it. It's a very small time frame, but it certainly can happen. Once you get past that 15th day, and progesterone is not even going to allow sperm to even get to where the eggs are. progesterone will stop up the cervix in preparation for complete pregnancy. So after day 15, forget it. If the eggs are not ovulated by then, and no contact's been made by then, it's not going to. So hopefully that answers your question. There's all kinds of crazy, phenomenal things that go on out there. I've heard of situations where she's 2 months pregnant, get pregnant again. That's not scientifically possible. But if you read the National Enquirer, there's all kinds of stuff in there. Now, is it the truth? Well, that's a good question. All right. Here's the last thing in our notes before our first test. We're prepping up for a test. We said we was gonna take it Tuesday, is that right?
No, I thought we said we was gonna do it Thursdays.
We're gonna try to do Thursdays, but I think if we did these notes and tried to review today, we'd be pressing the issue a little bit. So the first one's gonna be on Tuesday, and then after that, we'll try to set them on Thursdays. Good try though, try to kill a week. So we'll go over this today, and then Thursday, I'll review over your study guide that you're gonna have 99% completed by Thursday. If you have any questions, you need to complete it so you develop questions from it. And then I'll probably start some genetic lecture Thursday, because our next lab will be genetics, and I need to get ahead on that a little bit. If I lose Tuesday, then I'll just have next Thursday to get more genetics before we go to lab. But that'll be enough to get us started in lab anyway. It'll work out all right. All right, so the last thing we're gonna talk about here is over cell respiration. And cell respiration, this is something that happens in individual cells in the body where they take one molecule of glucose and produce tons of energy. I mean, every one of your little cells are little entities of you. They're all living. And so each one needs their own energy supply. And so we mentioned mitochondria the other day as your powerhouse. This is one right here. So we're taking one molecule of glucose, and we start through 3 stages. The first stage is glycolysis. Now, glycolysis, notice it says it's happening in the cytoplasm. It's out in the cytoplasm of the cell. And we have to have oxygen. Aerobic means with oxygen. Can we function anaerobically? Yes, for a short period of time. Trained athletes can function for longer periods of time. You can train your body to do more. If you're an untrained athlete, walking down the hall becomes difficult. Going up one flight of stairs, you're sucking air. That's how you know you're out of shape. So in this case, what we're ultimately trying to produce here is pyruvic acid. We're trying to take this one molecule of glucose and start breaking it down into substances the cells can use. So let's take, for instance, everybody's New Year's resolution is to get in shape after, first day of the year. Now that's after we've ate 14 turkeys over Thanksgiving and Christmas and the 10 hams and whatever fixings was thrown in there. I'm not even gonna talk about grandma's pies. We're feeling guilty. We wasn't when we ate it, but now we're feeling guilty. My knees is telling me I've gained some weight. So the first day of the year comes along. We're gonna get in shape, right? And you realize it's New Year's Day, isn't that a holiday? Let's wait till tomorrow. We gotta eat black-eyed peas and hog jowl today, right? So, second day of the year comes along, and we're gonna dive into this regimen. So we do some muscle stretches and some calisthenics, and we're good to go. And now we're gonna go jog a little bit. And so we just go ahead and jog us 5 miles. Don't overkill it the first day. Me, I get 100 yards down the road and I'm sucking wind. And finally, you ever get that stitch in your side? Just can't get that second? Well, that was me at 50 yards down the road. Got the stitch in my side. Well, what that is lactic acid. It's starting to build up in the system. Respiratory rate, your respiratory system's thinking, what are you thinking? I mean, that's what it's, what are you thinking? You're trying to do everything here, and we hadn't prepared for any of this. And so your respiratory rate's trying to catch up, and you're sucking so much wind, the weeds are coming out of the ditch, and you just can't get enough oxygen. So here we are, we're bound and determined to do it, and you get out there at 100 yards and you get this muscle cramp right here in the old hamstring. But you're determined, you're in pain, but you're determined, so you're dragging a leg and you're still pumping. Well, that didn't take very long until the other one cramps up and you just kind of roll over the ditch. All right, so what we've got here, we've produced lactic acid. We've produced a lot of it. That's what's caused your muscle cramps. Why do we have lactic acid? Because we have insufficient oxygen. Your respiratory rate has not caught up with it. In fact, your body just foreclosed on you because you didn't pay your oxygen debt. So when it foreclosed on you, it rolled you over in the ditch. Are you exercising now? You're just trying to stay alive. You're sucking air out of the weeds are going to and fro. But the good news here, there is some good news out of this. It won't be in the morning, but there is now. You get 2 ATPs. Now that is our usable form of energy. Adenosine triphosphate is what that stands for. And that's good. That's just enough energy to get your thumb up out of the ditch to hit your ride home because you're done. Now in today's time, it's not your thumb because nobody's going to pick up a bum in the ditch. It looks like you are right now. So hopefully you got your cell phone on you, right? And you got just enough energy to speed dial somebody you think might actually love you enough to come get you. While you're laying in the ditch waiting on them and they're taking their jolly time to come get you, they don't love you near as much as you thought they did. You're sucking air. How long after exercise do you breathe hard? Until you get your oxygen debt paid. In this guy's case, probably for 2 days. He wasn't very smart. Don't do this at home without consulting your physician. There's my disclaimer. And so finally, we get oxygen added back. Oxygen added to lactic acid changes to pyruvic acid. Okay? So once we hit pyruvic acid, respiratory rate starts coming back down. We get the oxygen debt paid. We get the pyruvic acid. You're allergic to exercise, aren't you? I am too. And we make it to the Krebs cycle. Wasn't that a lot of work just to make it? I'm tired thinking about that. Who's going to go home and start exercising tonight? Go buy you one of those Bowflex machines, right? Pay it off in 5 years. We finally make it to the Krebs cycle. It happens in the matrix of the mitochondria. I do like to ask where these things happen. So notice mitochondria. Cytoplasm. We only got 2 there. We're only going to get 2 here. But we take the pyruvic acid and we spin this cycle a couple of times and we get 2 more ATPs. But we get 2 molecules of this substance here, and I can't even pronounce that word, so we've got it abbreviated, PGAL. It is a very high phosphated compound, broke down from glucose. What I'll ask you out of this one is waste products. This is the cycle in which creates the CO2 and H2O. When you exhale, this is where it comes from, Krebs cycle. Every time you exhale, Krebs cycle produced your CO2, right here from glucose. And we always lose water when we exhale. So flip to the last stage. We're going to take these 2 molecules of PGAL into the last stage. So we take the 2 molecules into the hydrogen transport, which is also known as electron transport. The newest terminology is oxidative phosphorylation. This is phosphoration, but I just got target top in letters. And this is happening again in the mitochondria. This is in the Christie of the mitochondria. We take one of these molecules, PGAL, and ultimately it produces 17 ATPs. Since we have 2 of them, we double it, we end up at 34 ATPs. And I wouldn't worry about all these nidine diphosphates and flavidine diphosphates and stuff. Ultimately, we want to know ATPs, and this one produces the ATPs. So how many total ATPs we get from cellular respiration? Can you add them all up? 34 in this one. How many in the previous? Oh, maybe we got to go back to algebra. 34, you agree? How many we get out of Krebs? We just got 2. How many we get out of glycolysis? just enough to get her thumb up. 2. I'll have 2 fingers for you. 38 ATPs total. Now, the books will tell you that we use 2 to start the process over, so we're just gonna say 36 to 38 total ATPs. 36 to 38 total ATPs. Here's the type of questions you'll see from this. They're not hard. Where does it happen? Glycolysis happens in the cytoplasm. Krebs cycle in the matrix of the mitochondria. Hydrogen transport in the crystal of the mitochondria. Notice that 36 of the 38 ATPs comes out of the mitochondria. Is there any doubt now why the mitochondria is called the powerhouse of the cell? Tons of energy coming out of the mitochondria. Another question would be waste. Krebs cycle produces what? CO2 and H2O is waste. That's where your CO2 comes from. How many ATPs in each? 2 ATP in glycolysis, 2 in Krebs, 34, and hydrogen transport. So they're simple questions. I'm not going to ask you FADH and NADH, and I'm not going to ask any of that, but I do want you to understand that it's the mitochondria that's doing this. And every individual cell of the body is doing this. That's using glucose to do it. If you're diabetic, you don't have enough glucose, things shut down, don't they? Quickly. Any diabetics in here? Deal with it? Know somebody deals with it? You don't want to ever have to deal with it. It's a bad situation. Glucose, the brain's got to have its share, and that'll be the first thing that'll shut down. But now this is just a quick summary of what's going on inside the cell as far as energy production. And those are the simple questions that'll be asked from. You have any questions? Can we have the test Thursday? We do have a lab test Thursday, and it's over the tissues and mitosis. And we may do some questions on a couple other labs, take up some more lab time. So we won't have to do it down the line. They're not hard, but that's all we'll be doing.
Should we go there first?
Let's see, Thursday, review, come here first. Thursday, come here first. We'll do our lecture, then we'll go take our test, do a little bit of lab stuff, and we can go as you get finished.
Is there something like pictures we can study, or are we gonna go over before we take it?
Go online to my website, and the tissue pictures are out there, as well as the mitosis pictures.
Okay.
All right.